Welcome to the first episode of a journal walk. So what is a journal walk? You know, every day, let me cross the street. Every day I walk from home to lab and from lab back to home. And one way is to take about like 15 minutes. And I typically just listen to some music, but that's kind of boring, right? So then I decided, why don't I just take the time and doing a kind of vlog thing and to introduce a paper, just like what we did in Journal Club. Although the difference is, in Journal Club we have other people to talk about, but this is a journal walk and I'm walking alone, at least so far, and there's nobody I can talk to, so it's gonna be just a monologue presentation. Well, so today I'm gonna present or talk about the paper from Sarafyazd and Jazayeri 2009 science paper entitled Hierarchical Reasoning by Neural Circuits in the Frontal Cortex. So what is reasoning? I thought about this and I went to, obviously, Wikipedia, and it's a long entry, so I can summarize it in maybe one sentence. So reasoning is our ability to move from one side of ideas to other related ones, and with maximum likelihood. So in the extreme case, when the likelihood is, let's say, 100%, then that's sort of in the realm of like logical reasoning. So for example, like we see a man there covered in blood, and then there's another guy over there with a smoking gun, and the evidences are pretty obvious, and the man with the gun almost certainly just shoot the person in blood. But a lot of times we make this kind of inferences with uncertainty. So for example, like we know that a man was lying on the ground in blood, and then another one with the gun, but the gun is not smoking. Then we have to take the fact with grain of salt, and then to do the inference with this uncertainty. So, but you see, like here, like we have two ideas or facts. One is the person in blood, and then one is the person with the smoking gun, and then by combining the two pieces of evidence, whether they are certain or uncertain, and we can derive a conclusion. So things are pretty similar in this paper, except the one doing reasoning were two monkeys, and we have two different inferences. So although the authors here said they are hierarchical, but based on how I understand it, it's more like two parallel, separate inferences, but they are related. So the first one is what the monkey do during the task, during the execution. So what is the task? So for this particular inference is a rule-based interval discrimination task. So imagine there's a light flashing at the center, and then after a certain interval there's another flash at one side of the screen, and the monkey needs to determine whether that interval is shorter than a certain value, which is 850 milliseconds, or is longer than that. And in one setting, if it's shorter, then the animal succumbs to one side, and it's longer, the monkey succumbs to another side. But the side will change in a different context. In that context, when it's shorter, it succumbs to the opposite side, and then when the interval is longer than 850 milliseconds, it succumbs to the same side. So what is the reasoning here? So we see two pieces of evidence. So the first is whether the interval is shorter or longer, and of course this has an uncertainty in it, because the estimation of interval can never be perfect, and especially when the actual interval is pretty close to the 850 millisecond threshold. Well, the second piece of evidence is the rule, whether the animal needs to saccade to the same side or the animal needs to saccade to the opposite side given a certain interval. Here shows the psychometric curve that describes the performance of one monkey, and you can so the x-axis is the time relative to the threshold. Minus is shorter and positive is longer, and you can see, like, given one color, which is one rule, and there's a beautiful sigmoidal psychometric curve describing that the animal can do saccade or anti-saccade pretty reliably when the difference is big, and kind of more and more uncertain when the time interval gets close to the threshold. And so this is when the rule of the text is clearly given to the monkeys. But even if not given, the monkey was able to perform it reasonably well. So how do we model this process? So we can express this inference or decision making by expressing the action as the probability of, let's say, anti-saccade given what the animal believed or instructed to be the rule, and the actual interval the animal determines or perceives, which is TS. And clearly there are two parts. One is the perceived interval, and another is the role that the rule is playing. First, the author said the perceived time interval is follows a normal distribution with the actual interval as the mean, and omega times the actual interval as the standard deviation. So, and this really models something I believe called the Weber's rule. The uncertainty follows a linear relationship with the time interval. So the second part is the relationship between the action it's gonna take with respect to the perceived time and the believed threshold, the internal threshold, and the rule the animal believes in. So when the perceived time interval is shorter than the threshold, and when the rule is the second, then there is a probability 1 of doing an anti-saccade. And there are 3 other different combinations that either gives anti-saccade or not anti-saccade, which is 0. And the final conditional probability of doing an anti-saccade given the actual time interval and the believed rule, it's basically a marginal distribution, or you can say it's an expectation of doing an anti-saccade, given all conditions. Yeah, then that's the model for the first inference or the first reasoning. And then now let's move on to the second one. So there are 2 types of ways the animal can know or can figure out what rule it is. In the first type of experiment, the rule was clearly given at the beginning of each trial, so the animal doesn't need to figure out whether the rule has changed or not, because it's told clearly. In the second condition, there isn't an explicit rule given to the monkeys, and the monkeys had to figure that out through trial and error. And here we can see in this plot, the yellow curve shows the instructed condition where the animal immediately switched to the new contingency. The blue curve shows that in the inferred condition, in the condition where the animal has to refer the change of rule, it takes about 1 or 2 trials of errors before the animal can be confident that the rule has changed and change the behavior accordingly. And this second plot here shows that the probability of a switch or the change has to do with the task difficulty, with the difficulty of the discrimination. And when the discrimination is kind of close to the threshold, meaning when it's hard, monkeys were unsure about whether they made a correct discrimination or not, then they are slightly less likely to switch their belief in a rule when the actual interval is very different from the threshold. Then the animal will be very confident, and you see after one error trial, there's a pretty high probability of switch, and the probability goes even higher after the animal makes 2 consecutive errors. And of course, when there's no error, like the animal gets the reward, then there's very little probability of changing the belief in the rule. Okay, so how can we model that? So the author started by explaining the ideal observer model. So here is the value of Q, or is odd, and it's odd between that the rule has changed since the last rewarded trial and over the probability of the rule that hasn't changed since the last rewarded trial. And then of course we can like expand that in this way, which seems pretty complicated. And the 2 parameter here, like lambda, describes the like the hazard rate of the rule is going to be changed. Oh, I forgot to say, different rules are organized in blocks of trials, and how many trials in the block is determined randomly following a certain distribution. And the lambda here, which is the hazard rate that the experimenter used to switch from one block to the other. And what is the y? So y basically is from the psychometric functions, and it describes like the probability of correctly determining the interval, whether it's less than the threshold or larger than the threshold. But fitting a model like this requires a lot of data, so the authors decided to simplify the model using what they call the confidence-based model. And to start, the idea is pretty similar. So we have a quantity Q, and the Q again is a odd ratio of the probability when a latent variable x sigma is greater than an internal threshold theta, and over the probability of that variable is less than theta. So now like the decision is turned into accumulating evidence into this variable x sigma, and to see whether or how likely that sigma is greater than a threshold or below a threshold. When we only consider one error trial back, then we can equate this ratio with the previous ratio from the ideal observer model. And by rearranging the equation, and we can get that the by the way, the x sigma is modeled as by following a normal distribution with this mean and this standard deviation. So by rearranging the equation, we get this expression, and which can help us to, which allows us to solve for the parameters about x sigma. But x is a continuously changing variable, but the decision is binary, right? Like whether the rule is the first or the rule is the second. And so to turn that graded variable into a decision, and then here is this equation. So the threshold here is 1, which is the theta I mentioned. Alright, I have arrived at the lab. Actually, I have already been going around a little bit just for me to finish this slide. So when I going from the lab to home, I will finish the second part of the journal walk. Hi, let's continue. So I went to the lab at 1 p.m. in the afternoon, and right now it's 5 p.m. So 4 hours of experiment, and I got pretty good results. So that's the good news. So the author recorded from 2 brain regions. One is dorsal medial frontal cortex, and the other is anterior cingulate cortex. And the dorsal medial frontal cortex includes the SCF, supplementary eye field, and SMA, supplementary motor area, as well as pre-SMA, pre-supplemental motor area. Of course, the two most interesting things to look at to see whether the DM dorsal medial frontal cortical neurons and the ACC neuron encode is like the feedback of a trial, whether the animal gets a reward or not, or if the animal makes an error or not, as well as the switching behavior, whether the animal decided that the rule has changed and it needs to change its behavior. So the most straightforward way is to do some regression. We can model if a neural firing rate can be explained by a linear combination of the text parameters for each trial at each time point. And then if certain parameter is predictive to the neuronal firing rate, then you will see a bigger slope. And so therefore these plots on the axis are time, and on the y-axis is the slope, or here is called sensitivity because the authors normalize the slope across sessions and animals, I believe. So basically we're looking at some kind of slope on the y-axis. As you can see in DMFC, so these neurons encode the error signal at a very early time point. I think it's around 140 milliseconds or so. But in ACC you also see a little bit of that. But what distinguishes the two areas is whether they encode a switch signal. And for this you can only see that in an anterior cingulate cortex, but not dorsal medial frontal cortex. Okay, but we have spent a lot of time talking about models. We explained the model that that the animal may use to decide whether it's time to switch the switch the rule or not. We're gonna turn that problem into a key variable called x sigma, as well as the binary version of that is x yes or no. So then it makes sense to incorporate those two variables into this linear regression, right? Just add two other terms, well actually one other term, just x sigma, and put a coefficient in front of it. Yeah, and this was actually what the authors did as a first step, but the problem is obvious. The problem is even we get a pretty good slope for x sigma, but that doesn't mean like a lot of neurons are caring about it. Like we might have just selected a few neurons that's pretty informative or happens to be informative to this variable, but it doesn't reflect the population activity. This is the first problem. So the second problem is you can imagine like the linear combination of neuronal firing rate that reflects the x sigma may also help reflect the x yes and no. But it's important to know that whether like these two information can be encoded independently. So therefore the author used a method called the targeted dimensionality reduction method. So basically it relied on the PCA first to denoise the neural data. Denoising means like if you have like 30 neurons and then you use the PCA to reduce the dimensionality, discarding the components or dimensions that only has like very little variances, which likely due to noise. So the author only kept 12 principal components, which contains about 70% of the variance in the total population activity. And then like they projected the coefficients of x sigma and x yes or no to this 12-dimensional subspace. And once you're projecting to this subspace, and the noises will be gone, right? That sort of solved the first problem, whether the information was encoded by some random noise or a small amount of neurons rather than the main population dynamics. Although I will come back to this later, which I think is actually not guaranteed to achieve the purpose. It's better than nothing. Okay, so once we have that, now we're going to solve the second problem, which is whether encoding of the two variables, the two axes, are orthogonal or not. So here, for the targeted dimensionality reduction method, they basically orthogonize the two axes that encodes each of the variable by a QR decomposition. Okay, so let's look at the data first. Again, similar to the previous plot where we just show the sensitivity or the slopes of simple linear regression. So here we're plotting something that looks similar. X-axis is time, and it's time after the animal got the feedback, which is whether there's a... Okay, I'm already home, so but I haven't finished the journal walk yet. So why don't I just go to the beer store and buy some beer. So the distance is between the neural trajectory when the animal got the reward, meaning the trial was correct, and the second trajectory is the one when the animal made a mistake, and here includes both a single trial mistake as well as two trial mistakes looking back. And as you can see, X sigma is the greatest signal. Okay, so not sure if you can see this or not. The beer store is closed on Sunday, so I have to come over for the next time. So you see the components of neural activity that's predictive to X sigma doesn't really care about whether the animal switch or not in the next trial. So however, when it comes to X, Y, N, it's a binary resulting decision variables for the rule switch. So, and we see that the activities are really binary, and it only cares about whether the animal switch or not in the next trial, but not so much about the evidence, the accumulating evidence, which is reflected in X sigma. So, and you can see like the trend quantified down below here, or in these two plots. Okay, so here is my criticism about this method. It's not really a criticism, it's just a little complaint about it. I don't think this targeted dimensionality reduction method is very straightforward. I think it's quite a convoluted way that tries to do one thing but sacrificing another. Specifically, so you see it tries to denoise it, there's no problem, but when it orthogonizes these axes, the encoding direction is no longer the original or the optimal encoding directions, and it's less clear what that is going to be. They did PCA, and they use the neural activity across time as well as conditions of trials or trial types. But when they do regression, they did each time bin independently. So therefore, the variance for PCA and variance for the regression are really not the same, right? But that's okay. But I think they can be more consistent. And the last one, I hope they kind of report how much variance that the two encoding dimensions for X sigma and X yes and no actually can explain. Although I know that the 12 PC contains 70% of the variance explained, it is unclear once everything is projected to a specific subspace in there, and after orthogonalization, and how much each of them can still take. So therefore, I think there are other more straightforward methods they can use to serve a similar purpose. For example, the demixed PCA. So demixed PCA, I think, ensures orthogonality and is targeted to specific variables. So the source of variance is consistent, and it also tells how much variance is explained in a very straightforward and natural way. And although the mixed PCA might be better for categorical variables, like so here, for example, like the x yes or no may be better suited for this, but not as much as the graded variable here. Okay, so lastly, it's my favorite part of this paper, which is the manipulation experiment, where they stimulated a brain region and either look at what happens in the other region or what the behavior, how the performance changed. So the first experiment is stimulating DMFC while recording from ACC. And at the beginning, I wasn't very surprised or impressed, because we know like any area in the brain is more or less connected with any other areas. Oftentimes they redirect the synaptic projection, but like at worst, a few synapses away you get there. So stimulating one region is likely, or it's not surprising, to cause responses in another. So here they showed stimulating DMFC caused a slightly increased activity in ACC. But here's what's amazing. So they have two trial types, or two types of experiment that's conducted in the same session. So one is the instructed condition, where the cue is given to the animal, so the animal doesn't need to track the evidence whether the rule has secretly changed or not, because the rule is overt. And the other is the inferred rule condition. Animals did need to keep that in mind and track that in order to make the right decision about the rule change. And pretty amazing, like in the instructed condition, there wasn't an increase of firing rate in there, and the increase only happened when the animal needed to infer that. So the author interpreted this result as that ACC is downstream of DMFC. So therefore, and this connection, this pathway is only needed when animals performing the inferred rule task. And now let's move the stimulation electrodes from DMFC to ACC, and to see and still we stimulate the brain region just mildly without causing any changes in the motor control. And let's see how that affects the switch probability. Again, pretty amazingly, in instructed versions of this task, we don't see a significant change in switch probability, but when the animal needed to infer the rule, and we see an increased change of switch probability. And the two animals had this change in different ways, which you can see the figures yourself. I'll just skip that part. So overall, I really like this paper. I'm not an expert in this field, but luckily I was able to understand most of the method, and I appreciate how good experimental design is, and how good the data is presented, and how thorough their controls, both in terms of the behavior as well as the analysis, like those how well those controls were done. And of course, I certainly made some mistakes, misunderstanding, misreading some part of it. So please tell me when we meet and discuss this paper, or leave a comment down below. And also, I injected many of my personal views, personal opinions. So if I said something that you don't agree, also leave a comment. So lastly, I want to say thank you to Professor Daeyeol Lee. And without you, I wouldn't be able to know this paper. And so you basically send the paper out through email, probably to your lab members. And then I just indirectly got this paper from them. And the interesting side story is that I was writing some essay that's a little bit philosophical while talking about like the neuroscientific basis of moral philosophy. And I was writing about like our ability to reason, and is there any neural substrate for this ability. And then I just came across the paper you recommended, you disseminated, and I read about it, and I found that as a perfect citation to support my claim, or not really a claim, to support my argument in my essay. So yeah, and I'm probably also using the music as a background music, and I hope there's no copyright infringement that I did. So hope to talk with you in person sometimes. Yeah, thank you. That's it.